Alright, so I'm going to be doing my presentation on colorectal cancer. So overall, just to kind of talk about originally what I thought cancer was before I actually went and researched it a little bit more in depth. Um, I guess cancer to me was just a group of diseases that had a abnormal cell growth and it can be spread to other parts of the body, which is a little bit different than when compared to benign tumors, which don't really spread to other parts of the body. Some of the signs and symptoms would be like a lump or a mass, weakness, abnormal bleeding, prolonged cough, weight loss, change in bowel movements, etc. And there's obviously hundreds of different types of cancers that affect humans. So, kind of to talk about, I really wanted to focus on how diet really plays a big part in colorectal cancer. And research has really shown that eating several types of food and nutrients can play a risk in acquiring colorectal neoplasia. For instance, calcium, fiber, milk, and whole grain have been associated with lower risk of the colorectal cancer. And I'm actually going to refer to colorectal cancer as CRC. And then we have red meat and processed meat, which will increase our risk of getting CRC. There's a lot of evidence on the potential chemopreventive effects of vitamin D, folate in fruits and vegetables. And as a side note, CRC is the third most commonly diagnosed cancer and the fourth leading cause of cancer death in the world, actually. So prevention, like life and diet changes, are highly attractive and are educated in the community. Preventative medicine is the way of the future, even for asthmatics. I've reviewed quite a few articles, and I'll go over some of the dietary factors that may have a role in CRC, as well as some of the mechanisms associated with inflammation and immune dysregulation, given their role in colorectal carcinogenesis. So right here in my picture that I drew out, I'm really drawing the canonical and non-canonical inflammatory mechanisms relating nutrients to colorectal cancer. So consumption of saturated or trans fats increases free fatty acids and bile acid levels. Bile acids are metabolized by gut microbes to generate secondary bile acids, which can actually promote the colorectal carcinogenesis through increased pro-inflammatory effectors and oxidative stress mediated via the engagement of nuclear factor Kb and cyclooxygenase 2 pathways. Sulfur-containing amino acids and inorganic sulfur are also metabolized by gut microbes to produce hydrogen sulfide, which can be toxic to the colorectal mucosa and induce oxidative stress. As reactive oxygen species scavengers, antioxidant nutrients can protect colonocytes from oxidative stress, and B vitamins also counteract oxidative stress through lowering homocysteine. The calcium may inhibit colorectal carcinogenesis through direct effects on cell proliferation, differentiation, and apoptosis, and binding to free fatty acids and bile acids. Vitamin D exerts anti-inflammatory effects through bile acid catabolism, suppression of NF-κB, and COX-2 signaling, and enhanced production of interleukin 10. Omega-3 fatty acids can inhibit inflammation and promote resolution through lipid mediators and directly via the G protein coupled receptor 120 signaling. So here is pretty much like a little table that I drew out for calcium and vitamin D, which are really the two things that I wanted to focus on. Calcium is an essential for bone and dental health. Ionized calcium forms in soluble soaps with tumor-promoting free acids and bile acids in the colonic lumen, which is why we think calcium is anti-neoplastic. One article that I actually read saw nearly about 70% lower risk of CRC, comparing the highest and lowest quartiles of calcium intake. This promoted trials of calcium supplementation, and there was a big drop in adenoma. Other mechanisms through which calcium might affect CRC are inhibition of cell proliferation, promotion of cell differentiation and apoptosis, suppression of oxidative DNA damage, and modulation of CRC-related cell signaling pathways. This is probably mediated by extracellular calcium sensing receptor signaling, which contributes to the integrity of intestinal barrier function and homeostasis between gut microbes and immune response. Um, Something that I read in one of the articles that was really interesting to me is that it stated that the association between calcium intake and colorectal neoplasms may differ by anatomic location, with stronger associations observed for cancers in the distal colon or rectum. And anatomic site heterogeneity may be driven by microbial composition or metabolic variation in the colonic lumen. Calcium may have a differential effect based on the other dietary factors or genetic background. The inverse association between calcium and colorectal neoplasms may be confined in individuals with a high vitamin D level, reflective of the regulation of calcium level of vitamin D or low calcium magnesium intake ratio, due to competition between these metals for luminal absorption. So overall, most evidence shows a higher CRC risk among people with calcium intake lower than 700 to 1,000 milligrams a day. So we should really encourage people to increase their calcium intake to a level above this range. As far as vitamin D goes, I would say back in the 80s, there was a study done that proposed that vitamin D accounted for the high mortality rate of CRC in populations with low solar UVB radiation exposure. There's been studies that tested using this range of surrogates for vitamin D status, like solar radiation, circulating 25 hydroxy vitamin D levels, dietary and supplement intake, and predicted 25 OHD based on major determinants of vitamin D status. Most of the evidence supports an inverse association of vitamin D with adenoma with CRC incidence and mortality. One study suggested that the benefits associated with serum 25 OHD may require at least 75, with optimal levels between 90 to 100 for multiple health outcomes, including CRC. Some studies suggest an anti-cancer activity of vitamin D functions by binding to and activating the nuclear VDR. A polymorphism in VDR-BSML has been consistently associated with CRC. Vitamin D may directly or indirectly regulate 3 to 5% of the human genome and has been implicated in a wide spectrum of anti-cancer activities, like anti-proliferation, induction of differentiation and apoptosis, anti-inflammation, inhibition of invasion and metastasis, and suppression of angiogenesis. Among these, the anti-inflammatory and immune regulatory effects of vitamin D are really compelling and may mediate its role in vascular, neurologic, and autoimmune and infectious diseases. And one study actually researched mice with colitis, a CRC risk factor. Consumption of high vitamin D diet attenuated inflammation, which means that vitamin D may have a big part in carcinogenesis. So high plasma 25-OHD was strongly associated with lower risk of colorectal tumors with high lymphocyte counts. There's clearly an established role of vitamin D in immunity and the ability of immune cells to generate biologically active vitamin D, which gives us evidence for the importance of host immunity and vitamin D mediated CRC prevention. And overall, there was a lot of good data from studies that supported the potential chemopreventive effects of vitamin D against CRC development. There are still some trials being performed with vitamin D supplementation that includes cancer as a primary outcome. So hopefully there will be more information in the nearby future. Until then, we should really encourage and ensure that our patients obtain a sufficient level of vitamin D to maintain a plasma level of 25 OHD greater than at least 36 grams per milliliter. So, as far as this drawing that I did, it really shows the metabolism and anti-neoplastic pathways of calcium and vitamin D. So the vitamin D signaling the via vitamin D receptor, the VDR, and retinoid X receptor, which is the RXR, regulates the transcription of numerous genes involved in carcinogenesis through effects on inflammation, cell proliferation, differentiation, apoptosis, and angiogenesis. Elevated intracellular calcium together with diacylglycerol, or also known as DAG, results in the activation of protein kinase C, which is I wrote as PKC, and that can inhibit the cell proliferation and promote differentiation through multiple downstream signals. CAR signaling may also inhibit cell proliferation through inactivation of the beta catenin pathway. Vitamin D is an essential regulator of calcium homeostasis through the negative feedback system involving parathyroid hormone, the PTH. The secretion of PTH is enhanced as circulating calcium concentration decreases, resulting in increased synthesis of 1,25-OHD3. Elevated 1,25-OHD3 in turn promotes calcium absorption in the intestine and calcium release from bone, which actually suppresses the PTH production. So overall, that was just kind of really focusing on calcium and vitamin D. I really wanted to draw this picture though because I thought this is pretty cool. I did come across obviously a lot more information when I was doing some research. So even though I focused on calcium and vitamin D, I want to kind of talk about what I drew here, which is really just different kind of mechanisms relating foods in general to the colorectal cancer. So red and processed meat may contribute to CRC through generation of carcinogens, including the N-nitroso compounds, or NOC, heme iron, sulfur-containing amino acids, and saturated fats. Overweight or obesity, as well as chronically increased levels of insulin, may also have a critical role. Despite high saturated fat content and its role in increasing insulin-like growth factor 1, or IGF-1, dairy products may inhibit colorectal carcinogenesis via a variety of bioactive constituents. So calcium, vitamin D, conjugated linoleic acid, butyric acid, lactose and lactic acid, bacteria, and fermented milk products. Fruits and veggies contain various components with anti-neoplastic potential, fiber, antioxidants, B vitamins, and glucosinolates. Potential anti-cancer mechanisms of fiber are binding or diluting secondary bile acids, increased stool weight, decreased transit time, and bacterial fermentation, leading to a reduced colonic luminal pH and production of pyruvate, which has antineoplastic properties. Fiber, fruits, and vegetables may also influence carcinogenesis through decreased adiposity. So that's pretty much it. So I'm going to just kind of wrap up everything. So there is a few data from trials that support the efficacy of dietary modification or nutrient supplementation for CRC prevention. Several foods and nutrients that I found and read about that are related to CRC are red meat and processed meat, fiber, milk, calcium, and whole grains. They all have specific biological mechanisms derived from experimental studies linking them to CRC. And we should, of course, continue our research and investigation there into dietary strategies for CRC prevention. There's still quite a few unknowns that need to be addressed, like what is the optimal dose, formulation, time window for exposure to specific nutrients, foods, or dietary patterns to reverse carcinogenesis, or how do dietary components interact with the gut microbiota, as well as other host factors to influence CRC risk. These are only a few questions that still need to be answered, but in due time, if we continue our research, hopefully these questions will be answered fairly quickly, so we can really reduce the incidence and mortality for these patients. And overall, here are some of the references that I used for my presentation. Thanks for listening.